The Correa Lab at Rice University is collaborating with Moody Gardens Aquarium in Galveston, Texas. This past spring, we teamed up to set up a series of different field experiments in Moorea, French Polynesia, at the Gump South Pacific Research Station. My name is Lauren Howe-Kerr, and I'm a graduate student in the Correa Lab at Rice. Jake Emmert, the Moody Gardens Dive Safety Officer, and I used scuba diving to identify and tag individual coral colonies that are in an experiment to examine the effects of heat stress and nutrient enrichment on coral health. Corals harbor a variety of microscopic organisms in their tissue, and we're interested in learning about how these microbes affect how the corals are able to respond to stress. In order to do this, we collect small coral tissue samples from these colonies. Back on the boat, the samples are preserved for further processing in the lab. A typical day in the field includes going out on the station boats to collect the coral samples, bringing the samples back to the lab at the research station, and then processing these samples for different types of analyses. By preserving the genetic material in the coral tissue, we can examine which types of microbes are present in the healthy versus the stressed corals. This will help us understand different microbial mechanisms that affect coral reef persistence and influence how we might approach coral reef conservation issues. We will be returning to Moorea during 2019 to continue the next phase of these experiments.